So our inspiration for conducting our work basically looked at the idea that although as we all age, we build up free radicals and increase oxidative stress in the eye, not everybody develops age-related macular degeneration. So we came up with a hypothesis that it's potentially an inflammatory component of the disease that decides which person develops the disease versus which person just go through an aging process without any problems. Right. So we decided to look at different genes that could be delivered into the eye with the idea of dampening the immune response in these patients down the road. So we developed a in cell culture method to validate our hypothesis, and then we moved forward with the in vivo testing in animal models of inflammation to validate that our vector would work in vivo.
Henrique Jaime, why did you choose the myxoma virus specifically?
Okay, so the protein that we're using comes from myxoma virus. It's a pox virus, and this viral protein inhibits inflammation, and it actually works on two different pathways, the inflammasome and NF-kappa B. So we want to use that protein as an anti-inflammatory therapy. So first we tested it on cell cultures to see if it would actually block inflammation, so we looked at cytokine levels, and then we made a secretable version of the protein, and we looked at doing a transfer of the media of cells that were exposed to the gene, and then transferring that to cells that haven't been exposed, seeing if the biological activity persists. So after we looked at the cell cultures, we transferred to animal model. So we did a short-term endotoxin-induced uveitis, looked at inflammation there, also looked at the cytokine levels, and looked at the number of inflammatory cells that were present, saw a decrease in both.
What method did you use to deliver the package?
So we have this protein, and we made a secretable version of it, and then we packaged it into an AAV vector, and we used that for delivery into the vitreous of the eye.
And what were the results?
We found that first of all, in cell culture, our gene was able to decrease the secretion of these inflammatory signals, and then we moved into a transfer in vitro model that show that not only it can decrease inflammatory signals in the cell, but that function can also be transferred to other cells that were never exposed. Furthermore, we took this into an in vivo model using the animal uveitis model and observed that in that system, it also decreased the inflammatory signals and decreased the number of inflammatory cells, suggesting that this gene could be a potential anti-inflammatory therapy for inflammatory ocular diseases.
What are the next steps with regard to this project?
Okay, so we tested in a short-term animal model. We want to look at longer term and look at different diseases in animals that we could use to later translate to humans. So one of them that would be a goal would be age-related macular degeneration. So that'd be one of the bigger ones we want to look at in the future.
So what specific studies do you plan going forward here?
So first, we would like to conduct a long-term study in our version of geographic atrophy model. And this model, just like the human diseases, it starts with increase in oxidative stress, and it progresses in a slow fashion towards the development of geographic atrophy-like phenotype. So we would like to test if by intervening early in the disease with our anti-inflammatory vectors, we can alter the phenotype that we observe in this mouse model of the disease. We're also interested in testing in different models that have been that are available through different colleagues.
In terms of the timeline for your research, what is that likely to be?
We're potentially talking about several years between, my best bet will be 10, 20 years from now. We could probably have something that we can test for safety in human trials.
So what is your message to patients out there who are looking at what you're doing in terms of its translational impact? When do you anticipate that this might bear fruit in the clinic?
Based on the current state of the medicine practice, it would take a significant amount of time. But as new techniques are available and new information is available about these genes which are being studied in for other purposes, we can actually expedite the process in a fashion. But in the meantime, I think it holds promise for potentially stabilizing the disease and giving a hope for extra years of vision to these patients.
Thank you very much. Jaime, do you want to add something?
I think he cleared it up pretty well. I thought we want to go into the future and look at longer term inhibition of inflammation to see how we can save the vision future.
Anything you'd like to add to that?
No, we're very happy to be part of the young cadre of researchers that are trying to improve the vision in patients, and we hope that our work can bear fruit and bring in something new to the clinic for those people in need.
Thank you very much. This is Dr. Dave Seftel reporting for Macular News from ARVO 2014.